Chapter 17 deals with the cell cycle. So the cell cycle is divided into 4 different processes. The first is cell growth, then DNA replication, distribution of the duplicated chromosomes to daughter cells, and then finally cell division. So you'll notice in this particular lecture I've got things in red, which you definitely want to know for your test. So the cycle then will have an M, G1, S, and G2 phase. So the M phase is mitosis. That's where nuclear division is going to take place, and will usually end with cell division or cytokinesis. Interphase is a period between mitosis divided into G1, S, and G2. So the G1 is a gap phase, and that's an interval between mitosis and DNA replication. So it's active and it's growing at this time. S phase is when you actually have the synthesis, so DNA replication takes place. And the second gap phase is where cell growth continues, proteins are synthesized in preparation for mitosis. Now, in some cells in adult animals, there is no cell division at all, and nerve cells are an example of that. Others may divide only once in a while just to replace any cells that have been lost. So analysis then requires identification of the phases, and basically the phases of interphase must be identified biochemically, and that's usually by how much DNA content is there. In animal cells, G1 are diploid, which means there's 2 copies of the chromosomes. Their DNA content then is what we call 2N. S phase replication then is going to increase the DNA content to 4N. And again, the content can be determined by incubation of cells with different types of fluorescent dyes that bind to DNA. A really cool activity, guys, or type of instrument that you can use is called a flow cytometer or fluorescence activated cell sorters. And if you take a look here, guys, you can actually count the number of cells, and as the number goes up, here's your G1 and then the amount of DNA per cell, and then it goes down, you've got your synthesis taking place, then it goes up when you have G2 and M. So a flow cytometer is used then to measure this. Now, progression of cells through the division cycle is regulated by a lot of different signals. I know we just got done talking about cell signaling, but there's a lot of different signaling that takes place for different reasons in your cells. So these processes, such as growth, replication, and mitosis, are regulated by a series of control points. So please know that a major control point is called the start point, and that controls then progression from G1 to S. That was first designed or developed and recognized in yeast cells. So basically, here's your start site. You've got your nutrients, mating factors, cell size. Things start to go around. If you take a look, here's your yeast is budding off like that. Eventually, the daughter cell busts out, and then the cycle can continue. Now, in most animal cells, we have what's called a restriction point in late G1, and that's like the start point that we saw in the yeast. Now, passage through that is regulated by extracellular growth factors, and we'll talk about just a couple in this chapter. If the appropriate growth factors are not present, the progression stops at the restriction point, and the cells enter what we call a resting stage. So G0, please know that, guys, are a resting stage. So skin fibroblasts, for instance, are arrested in G0 until they are stimulated by platelet-derived factors to proliferate and repair different types of wounds. So again, here's your restriction point, and unless you get past that, you kind of are in this quiet time of not doing much of anything. So coordination then of cell cycle phases is dependent on a series of cell cycle checkpoints. So there's cell cycle checkpoints. So we have DNA damage checkpoints, Those will ensure that the damaged DNA is not replicated and passed on to daughter cells. Then we have spindle assembly checkpoints. That stops mitosis at metaphase if chromosomes are not properly aligned on the spindle. So again, if you take a look at our cycle, we've got the spindle assembly because that takes place in mitosis. This one, and this one actually will recognize if there's DNA damage taking place and stop the cycle at that point. Now, we do have different types of proteins and enzymes here, and we do have protein kinases that will trigger the major cell cycle transitions. I'm not going to have you know the whole list, so please don't worry about that. Okay, so what you should notice here, guys, we have cyclins in here. CDK1, that's a type of kinase that I'll mention in a minute. And what you're going to see, guys, is you have a series of phosphorylations and dephosphorylations. And when you get to certain points, you may shift over to mitosis, or you may just continue the cycle going this way. Now, remember we mentioned ubiquitination. Now, that's an important part of our cell cycle. And if cyclin B gets ubiquitinated, guys, it's actually mediated by ubiquitin ligase. And what happens then, it gets activated as a result of phosphorylation of the CDK1 and cyclin B. So a lot of research has gone in, guys, and it's been established that the CDK1 and cyclin B are really important parts of the cell cycle. Red, red, red. So the CDKs, CDK stands for cyclin-dependent kinases. So there's a lot of the cyclins and a lot of cyclin-dependent kinases out there. So the CDKs are regulated by 4 mechanisms. If the cyclin-dependent kinases and cyclin come together, then you get this complex that gets formed. And here's a good example of that. Now what this is showing here, guys, just so you know, are tyrosines and threonines that have been phosphorylated. And then binding of inhibitory proteins, or CKIs, so they're CDK inhibitors, can also take place. And in mammalian cells, there are 2 families of those. So defects in any of the cyclin regulation could contribute to loss of regulation and, again, can actually lead to cancer. So I'd like to talk a little bit about a tumor suppressor called RB. And RB, guys, is a protein or a substrate protein of cyclin-dependent kinase 4 and 6 and cyclin D complexes. And it was first identified in retinoblastoma, hence RB. And that's a rare inherited childhood eye tumor disease. RB is what we call a prototype of tumor suppressor. There's another one I'll talk about in a minute. And what it does is it inactivates tumor. If it gets inactivated, it leads to tumor development. So again, there's different proteins that play a role in there, guys. But if you think about it, if it normally suppresses a tumor, if you inhibit it, then the tumor is no longer suppressed. Okay, so if you suppress a suppressor, then you actually get activation. Okay, so I'm not going to hold you responsible for everything that happens in here, but we do have RB that binds to a transcription factor and then suppresses expression of the genes. So here's your factor, and then phosphorylation, again, that's a kinase. It will phosphorylate that, and then transcription gets activated. Now, if this gets mutated, guys, and stays on top of there, you can't get transcription to occur. So at DNA damage checkpoints, guys, the cycle arrests at the DNA damage. And this is mediated by 2 proteins, ATM, not that kind, ATM and ATR. And they then activate a signaling pathway that leads to cell cycle arrest. So please know that ATM recognizes double-stranded breaks and ATR recognizes single-stranded breaks. And then they phosphorylate and activate the checkpoint kinases CHK1 and 2. So again, ATR single-stranded, ATM double-stranded. These get phosphorylated, and that's how the regulation then occurs. So another tumor suppressor, guys, is P53. It's technically a transcription factor that increased levels will lead to induction of CDK inhibitor, what's known as P21. So again, what it does is it regulates the cell cycle in such a way that if there's any kind of damage in DNA, P53 stops it. However, guys, if P53 happens to be mutated, this actually, for whatever reason, increases the amount of transcription, and this is something that happens in cancer cells. And P53 actually is a gene that I had to sequence for my master's degree years ago. So again, it's frequently mutated in human cancers, and loss of P53 will prevent then cell cycle arrest, and then the cells just continue to replicate with this damaged DNA. And just some kind of cool pictures, guys, to show you to end.